What is neuroscience? Neuroscience combines biology and psychology with the aim of explaining behavior and understanding diseases through biological mechanisms. Neuroscientists want to answer questions such as, how does a disease affect test performance? How do medications affect test performance? And how do genetics affect test performance? The running wheel assay is a measure of locomotor activity in animal models and involves observing how much a subject runs in a wheel or on a small treadmill. The running wheel assay can be used to answer such questions as, how does the subject move? How much does the subject move? What happens in the brain after exercise, which can be figured out by using biological tests in conjunction with assays of this sort? And how long does the subject remain engaged? Some of the brain regions involved in this assay include the motor cortex, which controls movement, the striatum, which is important for motivation, movement, and coordination, the brainstem, which is responsible for balance and movement, and many more. Here you can see the setup for the running wheel assay. It typically features a running wheel inside of an isolated metal box. Here's an example of a rat performing a running wheel assay on a treadmill. The sucrose preference test measures the degree to which a subject prefers a sugary drink to water. This test can provide information about anhedonia, which is a loss of pleasure or interest, because normal animals prefer sugar water, while depressed animals lose this preference for the sugary drink. The test can also be used as a secondary measure of sensory function. You can ask the question, is taste sensation intact? The parts of the brain involved in this test include the striatum, the prefrontal cortex, and the thalamus, which is a sensory relay center. Here's a diagram showing the setup for the sucrose preference test. In an isolated box, 2 drinks are provided in 2 separate containers. The rat is allowed to choose between either of the containers, and the preference for one drink over another is measured as a function of how much one drink is consumed compared with the other. Here's an example of a rat performing the sucrose preference test. The rat starts off by drinking the water, then tries the sugary red drink, and as you can see, the rat will end up preferring the sugary red drink to the water.
The open field test is a behavioral test in which a rat is placed in an open box. This test provides information about locomotor activity and anxiety based on how much the rat moves and what areas of the box the subject explores. The test shows involvement of a number of brain areas, including the motor cortex, striatum, and brainstem, all related to movement, and the amygdala, which is related to fear and emotion. Here, you can see a diagram of an open field test. It is much like a box. This is an example of an open field test. In this trial, the rat is hugging the walls in an effort to protect itself, indicating that the animal is anxious. The radial arm maze is a behavioral test in which arms extending from a blank circular chamber are all empty except for one arm, which contains a reward. The rat performs this test multiple times in an effort to return to the same chamber and find the reward more than once. This test provides information about spatial learning and working memory, and shows information about the hippocampus, motor cortex, striatum, and brainstem. This is a diagram of a radial arm maze. Here, you can see an animal going to a chamber where she finally locates the reward.